Welcome everyone for the colloquium by Professor P. Balaram. And of course, Professor Balaram doesn't need any introduction, but for the benefit of the younger colleagues and students, I must state some words about him. So, Professor Balaram was, of course, at IISc Bangalore. Currently, he is a chair professor at the National Center for Biological Sciences in Bangalore. He obtained his bachelor's from Ferguson College in Pune, and then he went to IIT Kanpur for his master's in chemistry. And then he went stateside and did his PhD from Carnegie Mellon University in Pittsburgh in the United States. And during his PhD, he worked on negative nuclear overhauser effect. And I think that was probably the first demonstration of negative NOE using NMR. And after a very quick PhD, he moved to Harvard in 1972 to work with Robert Woodward as a postdoc. And of course, many of you know Robert Woodward is a highly celebrated chemist. And during the time Professor Balaram joined as a postdoc, he and Eisenhower had just accomplished the epic synthesis of vitamin B12. And Professor Balaram went on to work on another molecule called erythromycin, which, of course, many of you know has fairly complex and esoteric chemical features, including 20 stereocenters. And that was the work Professor Balaram was involved in during his postdoc with Robert Woodward. And he returned to India in 1973 and joined the Indian Institute of Science, Bangalore, newly formed department called the Molecular Biophysics Unit, headed by G.N. Ramachandran. Of course, before returning to India, Professor Balaram had met G.N. Ramachandran at the University of Chicago. And of course, he narrated his interesting story about his first interaction with G.N. Ramachandran in Chicago in this place 5 years ago at a keynote lecture at a meeting I organized on intrinsically disordered proteins. And in the early days in Bangalore, Professor Balaram was involved in various fascinating aspects of membrane active peptides, lipids, and bile acids. And he was interested in a very unique molecule called aromaticin, which is essentially a peptide antibiotic produced by fungal, of fungal origin. And this molecule contains an unusual amino acid called aminoisobutyric acid, or alpha-methylalanine. And of course, this is an unusual non-standard amino acid. It's not taken up in proteins, but it's taken up in peptides. And then, of course, at that point, he started his long-standing research on AIB, or aminoisobutyric acid, that promotes helix formation, various kinds of helix like alpha helix and 310 helices in peptides. And much of his early work actually occupied the study of AIB. And of course, I understand that many students from my chemical biology class are here. I really encourage many of the students to go back and take a look at his remarkable articles and accounts of chemical research in 1981, which has some of these aspects. And then he was also interested in fascinating aspects of D-proline that induces beta-hairpin structure in peptides and polypeptides. And then also he worked on beta amino acids, gamma amino acids, giving rise to exquisite structures, including hybrid helices and so forth. And later on, of course, he became interested in proteins, and he worked extensively on triphosphate isomerase from Plasmodium falciparum. And it was during that time I was doing summer internship with Professor Balaram in 1998. And that was the time when the IISc also got the first electrospray ionization mass spectrometer. And Professor Balaram's research then, of course, took a inflection into mass spectrometry of peptides and proteins. And in addition to NMR, circular dichroism, infrared spectroscopy, he was also quite interested in mass spectrometry, which is still pursuing at NCBS. So I have to also say that his seminal work has been very widely recognized both nationally and internationally. And the most recent one being the prestigious Bruce Merrifield Award from the American Peptide Society just a few months ago, which is a very distinct recognition, first recognition from India. And of course, as you can imagine, and during his long and illustrious and prodigious career, has produced some really terrific students who are now faculty, both in India and abroad. And of course, he has also been quite involved for many years in a journal, an Indian Academy of Sciences Bangalore journal called Current Science, as in his capacity as the editor-in-chief. And during the time he was the editor, he wrote some influential and incisive articles on various academic matters. In fact, I remember when I was a student at IISc, many of us would run into IISc library to read the latest current science issue, to read his editorial. And it was really, good old days, and we were really, we enjoyed reading his editorials in those days. But I must also add that apart from his, towering scientific contributions, what really distinguishes him is he has a deep philosophical perspective, which have been quite fortunate to have observed for the last 25 years, first being as a student and then being as a faculty. And his very, clarity on various matters, and clarity in thoughts, long-range vision, and also critical assessment on various academic matters, which I think is very important as a scientist, and we have all learned from him. So you can imagine that it's really a great occasion, and we are hugely honored to have Professor Balaram here. And I would just like to show you one photograph of Professor Balaram when he joined, right after he joined IISc Bangalore. He joined IISc Bangalore in 1973 at the age of 24. He joined the faculty at the age of 24. And from grapevine, I had come to know from my good friend, late K.S. Krishnan in TIFR, that he has the distinction of being younger than his first PhD student. So I must show this epic photograph, which I got from Professor Manju Bansal's article. And you can see in this photograph, Dorothy Hodgkin, which I've talked about in my chemical biology course on various occasions, and G.N. Ramachandran, who hosted Dorothy Hodgkin in 1974. And of course, there are many others, C. Ramakrishnan, whose work also have highlighted. And of course, in case you are unable to, of course, this is Professor Manju Bansal. And in case you are unable to spot Professor Balaram, here he is in 1974. And we are indeed honored to have Professor Balaram. Professor Balaram, over to you.
Thank you for that rather long introduction, Samrat, and also for giving away my age to the audience. And, I must say one thing. I'm comfortably and happily retired for quite some time now. It's a great pleasure to come back and be able to talk to so many of you once again. Now, over the last 2.5 years, uh, our lives have been dominated by the coronavirus and the pandemic. And so I thought what I would do in this lecture, which is somewhat general, to talk about what I have done during the coronavirus pandemic, what have I learned about the coronavirus, and also to reflect on the two disciplines in which I've been interested, chemistry and biology. Samrat also wanted to check out whether I still am good at doing what I'm supposed to be doing. So tomorrow morning, I will give a seminar which is distinctly more technical than the one that I'm giving right now. What I'm going to tell you is an abridged history of the coronavirus, little biology, a very little chemistry, and some reflections on the evolutionary history of the coronavirus spike protein during the pandemic. Now, the slide is decorated with the two foundational pillars of chemistry and biology, Mendeleev on the left and Darwin on the right. So you can draw your own conclusions from the figures that I have put there. Now, very often when you are a spokesman for science and generally trying to promote science in colleges and other places, and as I've gotten older, I get called more and more frequently to talk to students generally about science. And I've always wondered, how do you define science? What is science? And science, of course, is the study of nature. Then, of course, the next question that you will be asked is, what is nature? And the answer that I found to this question was in volume 1, number 1 of the journal Nature. Those of you who are students here, all of you will want to publish in two science journals. One is called Science and the other is called Nature. Very simple names. In volume 1, 1869, number 1, the new editors of Nature commissioned the biologist Thomas Huxley to write the editorial. Huxley did not write the editorial. What he did was he translated from the German something that had been written by the poet von Goethe, who said that nature, we are surrounded and embraced by her, powerless to separate ourselves from her and powerless to penetrate beyond her. You can't say it better than that. And of course, the coronavirus was a force of nature. Now, when a force of nature strikes us, then of course, disaster happens. And in this case, of course, you see plots like this. This is July 1st, 2021. You can make one for July 1st, 2022. As the pandemic has raged, people have been infected, large numbers of people have died all over, and therefore, one really needs to understand what is this force of nature that we have encountered. So when the pandemic began, and I think all of you will remember this, in March of 2020, in the third week of March, we went into our first major lockdown. That was a great experience, I would say, for everybody. Here we were, locked into our houses. We were not supposed to go out. The Prime Minister had said that the front door of your house is the Lakshman Rekha, you cannot cross it, and therefore we shut our doors and stayed indoors. At this point, having nothing to do, I began to ask myself a question: what is this coronavirus? And one thing that someone of my age can do is to pick up the telephone and call other people and ask them the question. They'll always like to humor an old man, so they will answer you. I found something rather interesting. Most of the people I called didn't know what the coronavirus was, and they were biologists and they'd never heard of it. So then I realized that maybe we should find out something about the coronavirus. There were also some people who I thought were virologists, and they didn't seem to be completely familiar with this virus, wherever it came from. So I began to search the literature, and I came across this wonderful definition of a virus in this book, which is called Aristotle to Zoos. It's a philosophical dictionary of biology. That's its subtitle. written by Peter Medawar, one of the best writers of science in the English language and a Nobel Prize winning immunologist. And what Medawar said was, a virus is a piece of bad news wrapped up in protein. You can't find a better definition of this. After all, there's a genetic material, which is the bad news inside, tells the virus how to replicate. It's covered up with protein, and then of course it has to get into a cell, replicate, and do more damage. The only difference, of course, is that the coronavirus is an enveloped virus, so it also has a phospholipid bilayer. So if we move on and look at what the coronavirus would look like, that's what the Center for Disease Control in Atlanta has given us as a readily usable picture of the coronavirus. A nice spherical object with the spike protein symmetrically dotting its surface. Little more detail. Here is the bilayer membrane, the coronavirus spike protein embedded in it, and then of course the interior proteins of the virus inside, including the nucleic acid. So that's what it looks like. Over 100 years ago, one of the founding fathers of modern medicine, William Osler, said this. He said, soap and water and common sense are the best disinfectants. During the pandemic, we had plenty of soap. In most places, we had enough water, although there were some places in Bangalore which did not have enough water. But what was lacking throughout the pandemic, of course, was common sense was in extreme short supply. So now we've got to study the coronavirus. The Prime Minister, having declared the coronavirus as an enemy, also used this wonderful analogy of the Mahabharata, where he said the big war ended in so many days, and so we will in fact vanquish this virus in so many days. So if you're going to declare the virus as an enemy, we must follow this advice. Very good advice given by Michael Corleone in Godfather 2. He says, keep your friends close, but your enemies closer. It's extremely good advice for everybody, including administrators. Now, and I have been one, so I should know. So the best way to find out what the coronavirus is to start asking the question, who discovered the coronavirus? And I wasn't the only one asking this question. The BBC was asking this question, the National Geographic was asking this question, and they very quickly put up this picture of this lady here, June Almeida, who is the electron microscopist who took the first electron micrograph of the coronavirus, which I show you here. So when I saw this article in the BBC on the internet, I immediately went and picked up this paper. Appeared in 1967, famous virologist David Tyrrell, who worked on rhinoviruses causing the common cold, and Almeida and Tyrrell published this paper. Now, it says that they've characterized human respiratory viruses that grow in organ culture. And here are the respiratory viruses they characterized. Don't worry about the abbreviation. Just remember, this virus is called 229E. So that's the picture they have taken here. When you read a paper, and this is for the students here, and especially papers in biology today in journals like Science, Nature, and Cell, don't read the introduction, don't read the conclusions, because that's where the authors tell you that they've solved every problem that is to be solved in biology. Instead, you read the results, look at the figures, and read the legends carefully, and if you can, go and look at the supplementary material, because that is where most of the useful information is really hidden. But in the old days, the papers were simpler. So I read it carefully, and so there's figure 1AB, these are the pictures, and they say that this is because of the strain, they got this from the strain 229E, and further up you say strain 229E of Hamre and Prochnow 1966, 1 year earlier. So my question then was, who is Hamre and who isolated the coronavirus? And then down here you say they thank Dr. D. Hamre for the 229E virus. So I launched at this time in April of 2020 an internet search for Dr. D. Hamre. And that's what I'm going to tell you about next. That's the 1966 paper now, Proceedings of the Society of Experimental Biology and Medicine, and there's a new virus isolated, etc., etc. And you can see now Dorothy Hamre. So when I saw this, I was very happy. I now realized that the person who first isolated the coronavirus was in fact a lady. And now, of course, you must look at 1966. Now, 1966, there were not many ladies in American science who were doing spectacular things. It wasn't a very friendly place for ladies to be. And therefore, I thought she must be quite unusual. I read the paper. Now, the paper was remarkable. She'd established that the virus was an RNA virus by showing that it was not inhibited by DNA synthesis inhibitors. She'd also established the size of the virus. Classical methods, 89 millimicrons, units today would be 89 nanometers. Remember that number. You know, we are also fascinated by the word nano, that we must remember nanometers, 89 nanometers. This virus has caused acute respiratory distress, and there are reports, case reports in the medical literature in the past. Occasionally people getting this virus have gone into the kind of conditions that we saw during the pandemic. So in 1967 paper of Tyrrell, 1 year later, they measure by electron microscopy. Remember, electron microscopy was in its infancy at this time in biology, and there it is, 80 to 100 nanometers. When I saw this number, I was really happy because Dorothy Hamre's number was 89 nanometers. So here it is, she's done a wonderful measurement. She must have been a remarkable experimentalist. Having nothing better to do, I also realized that I must find out more. When did the coronaviruses first come into the literature of science? This is the place where it appeared. In the journal Nature, not in a paper, not in a communication, but a group of virologists had written to the editor of Nature, and the editor of Nature did not print their letter. Instead, he wrote his own little note saying, a group of virologists have told me that they found a new virus, which they are going to call coronavirus. So 68 is when the coronavirus first appeared in the scientific literature. You can see her name here, D. Hamre. In 75, they named it formally. By that time, her name had disappeared. So I was very intrigued by this. I said, who is she? She discovered the coronavirus. Now she appears to have gone away. And by this time, we were already becoming somewhat, what I would say, comfortable with online meetings. Already people had started saying, can we have an online lecture? Now, when I give a lecture, I put Mendeleev and Darwin on my slide. I like to put the pictures of the people I'm talking about. So I said, I must have a picture of Dr. Dorothy Hamre. So I searched. I searched all the databases that I could. First one, of course, is just go to Google, type the name, and look at images. And when I do this, I found many young women who were called Dorothy Hamre. I found models, authors, other people, but I didn't find anybody who looked like a virologist working way back in 1966. So I searched PubMed. I found she had 40 publications in PubMed, listed between 43 and 72. And then I found that Google Scholar, I put all these together, and this is the image that I got. She was a productive scientist. She had a career which at least spanned 41 to 72, and there was no picture of her. This puzzled me. If she had done some wonderful work like this, I should be able to find a picture of her somewhere. So what I did then next was search the internet even further. Finally, I found a reference to her in an archives at Northern Arizona University. And so apparently her husband, I didn't know it at that time, had deposited some papers in Northern Arizona University. It was they were photographs. So I wrote off on the 29th of April 2020 an email to the curator at the Northern Arizona University asking them could they please check in their collection whether they could give me a photograph of the discoverer of the coronavirus. They of course said we're all locked down, so we can't go to the archives and open these files and look at them. But I think they were intrigued by the fact that someone from far away had asked them this question. And so they did in fact go, I think the very next day, and search the archives, and sure enough they found photographs which had never opened and seen before. And finally, I found her picture. And so I wrote an article. And I was so interested in writing this article that I wrote the article before I got the picture, because I thought I would never get the picture. And you might wonder, why are you doing this? I was intrigued here because here was a lady who'd got her PhD in 1939. By this time, American documentation is so good on the internet. I knew where she was born, I knew where she lived, where she died, everything. And now I was surprised, somebody in the years of the Great Economic Depression doing a PhD, and subsequently working post-PhD during the years of the Second World War. As a bacteriologist, she worked on penicillin, and penicillin had just been discovered. So she must have been extraordinarily good. And this, of course, is not my article. The young archivist, whom I picture here, whom I've never seen, she was in correspondence with me all this while. A most interesting time when we were in the lockdown, I would send a mail, and throughout the night I'd look to see if I got a reply, because here it was, we were both searching, and there's nothing like a search. And she then wrote an article finding Dorothy Hamre in a journal, in an archives journal. And she said that here it was, that I contacted her and so on. But the real most important impact of my work was that I discovered where she died. She died in a small town in Colorado called Ouray. So I wrote to the people in Ouray. They have a newspaper which I found. So I wrote to them and said, look, do you have any information on her? They said, no, we don't have any information on her, but we will connect you to the Historical Society. So they connected me to the Historical Society in the small town of Ouray. It has a population of 1,000, and it has a historical society. You can imagine, you know, in India we rewrite history, we imagine history, but we don't document history. So documentation is extremely important. And so I got in touch with them. It was a very interesting conversation. It turned out that the president of the Historical Society was a retired professor of organic chemistry. You know, you go and retire and settle in the mountains in Colorado. So I then introduced myself. I said, see, I also studied organic chemistry. I studied under Robert Woodward. He said, oh boy, you studied under Robert Woodward? Here I am, I'm going to answer. So they then reprinted my articles in Frontline in their paper, and this is probably the best citation that I've ever received amongst all the work. I was interviewed then, and when I was interviewed, they asked me, why are you doing this? You see, Ouray is there on Google Maps, Flagstaff, Arizona is over there, and so I told them that it is a tribute to the power of the internet that the road from Ouray to Flagstaff passes through Bangalore. It also seems to me that in some strange way, the virus has led us to its discoverer by turning the world upside down. I would not have done this search if the virus hadn't appeared here and landed us in lockdown. So the discoverers of the coronavirus, if I were to give a class, I would put these three pictures. Yes, David Tyrrell, June Almeida, and Dorothy Hamre. But why did the coronavirus catch us by surprise if it appeared in 2019? It turned out that in 1996, David Tyrrell, who led the work on coronaviruses and respiratory viruses in England, wrote this article in the textbook of medical microbiology, chapter 60 is coronaviruses. He said coronaviruses cause acute, mild upper respiratory infection, in parentheses, common cold, and with that, funders, researchers, everybody lost interest in the coronavirus. But in 2002, 2003, the coronavirus reappeared. It came from southern China, and the first case of coronaviral infection, SARS-CoV-1, was detected in the Vietnam French hospital. And on the 28th of February 2003, they requested help from the WHO. The WHO sent this remarkable physician, Carlo Urbani, who immediately, by the first week of March, realized that they were dealing with an extraordinarily infectious disease. He persuaded the government of Vietnam to impose extraordinary quarantine measures. Urbani contacted the symptoms on March 11th, died on March 29th, 2003. So you can see SARS-CoV-1 was terrible. 800 deaths, 8,000 infection, but contained immediately, didn't spread further. You might ask why, and that's the kind of question that one must ask as to whether the current coronavirus was in fact a somewhat milder virus, which did not cause much disease in most people, and therefore spread silently and quietly from Wuhan up into other parts of the world before it began to adapt by mutation. I digress briefly into chemistry because I said I would. You know, any place where you go to, especially if they're college students, and you ask them how many of you like mathematics, hardly any hands will go up, a little bit like this. If you say how many like physics, many will put up their hands. You say how many like biology, some will put up their hands. And if you say how many like chemistry, you won't see the hand at all. It will be almost like it's been amputated. And one of the reasons, of course, is the language. And Galileo is reported to have said long ago that mathematics is the language in which God wrote the universe. You might just paraphrase that and say that chemistry is the language with which nature wrote the book of life. You can see that I am somewhat irreligious, so I simply replace God by nature. The real problem in learning these subjects is the following. To learn chemistry, you need to learn the language of chemical structures, and unless you're familiar with them, chemistry seems completely unlearnable. So too is mathematics. Their symbols, equations, their relationships, and so on. You need to learn a language before you learn the subject, and they don't relate very easily to real life like physics or biology. But Arthur Kornberg gave the best definition of chemistry many years ago. He called chemistry the lingua franca of the medical and biological sciences. And Kornberg was the man who discovered DNA polymerase in the 1950s and fired, I believe, the first shot of the biotechnology revolution. So you must learn chemistry if you're going to do biology or medicine. And it's one of the sad facts of the Indian education system that chemistry is not important either in medicine nor in the study of biology. The only way chemistry and biology differ are in length scales and time scales. So look at molecules, simple molecules on the side, and look at their dimensions, and look at the large, more complex aggregate of molecules that you have in cells and organelles, and look at their dimensions. They differ in time scales, they differ in length scales, they differ in energy scales. But eventually, you can understand biology only in the terms of the complex chemistry that happens within cells. This cartoon sort of summarizes the importance of chemistry. There's this man solving a stock crossword puzzle, and he asks, what's a 9-letter word for biotechnology? The lady sitting by his side, very smart lady, she immediately answers chemistry. And of course, if you ask what is a 9-letter word for material science, you would once again say chemistry. So without chemistry, you can't really do anything. I don't know how many of you know this, but many years ago, the Bombay High Court, in a famous judgment, declared that steam is not a chemical. They were actually adjudicating on a case in which steam was being produced by an industry in Mumbai, and it was being piped into the next plot of land where another company was using that steam to heat up whatever they were doing. So they were selling steam. And the question was, should you apply sales tax at the level of a chemical, or should you apply sales tax at the level of a non-chemical? A non-chemical, the sales tax is higher, and the courts always rule in favor of the government, and therefore the court said steam is not a chemical. The Royal Society of Chemistry followed this judgment and immediately announced a prize of £1 million for anyone who will produce a material that's not a chemical. The £1 million is still available to you. In fact, the only people who produce things which are not chemicals are Tata Chemicals, which produces salt. And during the cricket matches, we have only, I think, one more match left now. Please watch that advertisement. Tata Chemicals will advertise a salt which doesn't contain chemicals. It contains 80 minerals. And then, of course, there is our own Patanjali, who will sell you a chemical-free phenyl. So this unfortunately is the state of chemistry in public perception. The foundational pillars of chemistry are on this slide. There's of course Mendeleev, who tells us diversity, Wohler gives us unity, and I'll show you on the next slide. I've borrowed a physicist, Boltzmann, who gives us the idea of dynamics, and in fact gives us the idea of the atom itself. And Pasteur, again, I think his more fundamental work was in chemistry, and although he is the founding father of microbiology, vaccinology, and many of these other disciplines. This is Wohler's letter to Berzelius, where he united chemistry in one way, the chemistry which is abiotic with the chemistry which is biotic. After having produced urea by this wonderful reaction of heating ammonium cyanate, he wrote to Berzelius saying, in a manner of speaking, I can no longer hold my chemical water. I must tell you that I can make urea without the use of kidneys of any animal, be it man or dog. Wouldn't you like to write such a letter to someone? Biochemistry's origins are in Buchner's classic experiments, where he crushed yeast cells and showed that minus the cell, the juice itself is able to ferment sugar. The chemist Bayer, of course, who was his mentor, said that this will bring Buchner fame even though he has no chemical talent. So this is where really the divisions between chemistry and biochemistry began, where one felt that one didn't have to know a great deal about the detailed chemistry to understand biology. But with this digression, I return to the virus. And if I return to the virus, you know, we've all been very self-congratulatory during the pandemic. We've been saying how wonderfully science has done during these 2.5 years. We've learned everything that there is to know, and we've learned it very quickly. And now, of course, governments will say, we know how to make vaccines. We'll make them in 6 months, 3 months, 2 months, whatever have you. Much of what we've learned in the years of the pandemic is because a great deal of groundwork was done between SARS-CoV-1 in 2003 and the COVID-19 pandemic. There was a 16-year interregnum in which many, many papers appeared which quietly were buried in the scientific literature. Here is one, for example, from which I've taken this picture. It estimates the particle diameter now using the technique of cryo-electron microscopy. And you can see here, 82 to 94 nanometers. When I saw this, of course, my regard for Dorothy Hamre increased even more, because she said 89 nanometers, you can't do better than that. Today we know everything about the coronavirus spike protein, its other components and so on. In these pictures, all of you would have seen science in March of 2020. The reason the structure appeared so quickly is you already knew how to get spike protein structures from the SARS-CoV-1 episode of 2003, and research had been done for quite some time. It was also known that SARS-CoV-1 uses the angiotensin converting enzyme as the receptor. Today we have everything. We have the full-length virus, the full-length spike protein, we have, except for the transmembrane domain, and we have the receptor complex, everything in this picture, for example. This is again an article from 2020. Now, what will we do with these atomic level structures? How much do these atomic level structures now help us to move towards, let's say, a therapeutic, or do they help in vaccine design, etc.? So when you talk about doing atomic level structures, you sometimes ask, is it of any use? And even government will ask you sometimes that question, is all this of any use, any practical utility? To understand atomic level structures of even large and complex substances, you can't do better than to emphasize the importance of atoms. And the best way to do this is to go back to Feynman's famous lectures, undergraduate lectures in physics at Caltech, where in volume 1, number 1, he will start with matter is made up of atoms. Here, you read this carefully, ask a question. He says, if in some cataclysm all of scientific knowledge were to be destroyed and only one sentence passed on to the next generation of creatures, what statement would contain the most information in the fewest words? It's an interesting question. He answers this question, I believe it is the atomic hypothesis or the atomic fact or whatever you wish to call it, etc., etc. The idea of atoms and molecules is important. You might also ask another question, what is the cataclysm that Feynman was worried about? The cataclysm that he was worried about in the 1960s was the spectre of nuclear war, which would annihilate everything. Today you will find, 60 years after Feynman, we are still worried about nuclear war and what would happen if there were in fact rather uncontrolled nuclear exchanges where everything would really be destroyed. Having said something general about chemistry, and you already know that the virus is a biological object, we can ask, does the virus belong to a chemistry department or does the virus belong to a biology department? And you will find if you go to the literature, if you go to a biology textbook, you will find viruses are excluded from the tree of life. You will find eukarya, bacteria and archaea all there, but you won't find viruses anywhere. So where do viruses fit in? You will find both views in the literature. Reasons to exclude the viruses from the tree of life, and also are people asking questions, do viruses exchange genes across the super kingdoms of life? After all, when you look at the human genome, it will show you signatures of the past interactions of human beings with viruses on historical timescales. A little digression into biology. Now, if those of you who are chemists or even generally biologists, especially of the molecular variety, you must remember that you can't do biology without understanding evolution. And there's no one better than Darwin to tell you this. And what he talks about natural selection, adaptation and evolution, which happens even at the level of molecules, as we will see in the coronavirus, he says there's grandeur in this view of life, with its several powers having been originally breathed by the Creator into a few forms or into one. Darwin is religious, and that while this planet has gone cycling on, according to the fixed law of gravity, from so simple a beginning, endless forms most beautiful and most wonderful have been and are being evolved. This, of course, celebrates the diversity of biology. The three pillars of modern biology are illustrated on this slide. I have evolution and Darwin on the right, Mendel and genetics on the left. This is the 200th birth anniversary of Mendel, 2022. And one must go back and celebrate Mendel at this point. How are they connected? After all, variation and selection are the themes of Darwinian evolution. Genetics, of course, is the transmission of hereditary characteristics from one generation to the other. But the generations vary, and so evolution takes place. How does this happen? It's linked by the chemistry of DNA. So I've got Oswald Avery here, who discovered DNA as the as the pneumococcal transforming principle, and discovered that information transfer in biology happens through DNA. And of course, Watson and Crick, whose double helical structure really tells you how information is transferred. So it's chemistry which links Mendelian genetics and Darwinian evolution. If you go to the human genome, for instance, 2000, that's when it was announced. So many eukaryotic genomes have now been done. We know a great deal of biology. I put this up just to tell you that you must look at these trees, especially if you're students. This actually tells you about the unity of biology, where on a tree you will find humans, chimpanzees, rats, mice, dogs, cats, cattle, everything. They're all sort of clustered fairly close together, all the animals. So sometimes when we fight endlessly about pigs and cattle, biology should teach us about the unity of nature. Lastly, I'll come back to something that during the pandemic I did do something mildly technical, beyond searching for photographs of ladies. And that is the small amount that I want to tell you about protein sequence variation by random mutation and natural selection, and we will take the SARS-CoV-2 spike protein as the example. But before we do that, I must show you this. This is from 1970, and this is a wonderful paper. You must all read it. It's called Natural Selection and the Concept of a Protein Space. This is written by the evolutionist John Maynard Smith. This is all of a page in Nature. There's no figure other than the one that I have shown here. There's nothing there except English text, so anybody should be able to read it. There are no equations of any kind. He describes mutations. He says, let's start with the four-letter word, word. Make a single mutation, D to E, we get the word war. We make another mutation, we get the word go. We make another mutation, we get the word gone. Make another mutation, we get the word gene. So we've actually gone from word to gene with single letter mutations. But during this process, we have not taken the word out of meaning in the English language. It's still a word which has meaning in the English language. And what Maynard Smith says is, this is how proteins have evolved. So you have proteins, you change their function slowly, but if they still remain functional, acquire a new function, that now becomes another sequence adopted by the organism. And but protein sequences is an infinite space. There's so many sequences, variable lengths, 20 letters in the protein alphabet, and so on. You must read this article written by Nobel Prize-winning chemist Frances Arnold, 2011. She writes, "The library of Maynard Smith, my search for meaning in the protein universe," because she's doing evolution in the test tube, bacterial evolution in the test tube. But in her abstract, she says, Borges' infinite and unsearchable library is reflected in Maynard Smith's collection of all possible protein sequences. What is the reference here? The reference here is to a short story written by the Argentinian author Jorge Luis Borges. This is all of the story, so anybody can read it. And what Borges writes is, if you have a library in which the secret of the universe is stored, this must be an infinite library. So he says, how will an infinite library be designed? And that's what he gives. He gives the prescription for designing an infinite library. Of course, if you go into the library and search for the meaning of the universe, you eventually end up like this. You will die and you will fall down this bottomless pit, because this is in fact an infinite library. Protein sequences really are an infinite space of sequences. Some sequences are functional, that's a finite set of sequences, and that's what biology uses. So now if you take the SARS-CoV-2 spike protein, it has 1,273 residues. That means if I write it in the one-letter code, it has 1,273 letters. It's a sentence which contains that many letters. Now at each position, if I can use 20 amino acids, the total space would be 20 raised to 1273, which works out to 10 raised to 1656. This, of course, is a number so large that we can't imagine it. Not even some of our country's most imaginative scamsters can think of a number this large. So it's much larger than anything that I've ever seen. And then, of course, the coronavirus spike protein does very important things. It binds to the receptor. It needs to be cleaved in the center before, as a prelude to membrane fusion, and then virus internalization. Now, of course, the 1,273 letters are what I've shown here. This is the structure of the protein, and this is how the virus will come to the cell, contact the receptor on the surface, and then everything else will happen. Now, when mutations happen, all these processes may now be affected. Now, it turned out that when I began to look at sequence data, I had no access to Indian sequence data. Indian sequence data is generally secret because I think the government believes that if you keep things secret, it's good for the country. And now, of course, just a few days ago, the sequences have become publicly available. And they are now available in Indian Biological Data Center, ironically of which they appointed me as the chairman of the committee. So I now managed to get the Indian data, and have just begun to look at it. But you can look at the American data, and the American data is very good because both India and America did wonderful experiments. They have large populations, most of the people won't listen to anything. Actually, we were somewhat more disciplined, and in India, of course, most of the people can't listen for various reasons. In America, most of the people won't listen because they don't want to listen. So you have the virus now floating around in the human population, mutating away, and you can now go back and look at all these sequences. With the help, I can't do all of this. That also you should know. Professor Joshi, who is a retired colleague of mine, an ecologist, a statistician, but very good with computers. For the benefit of Professor Rudra Pratap, who's here, I might say he's still a person who writes his own code and works with Linux machines. So you can't beat anybody who does that. So you can do many, many kinds of analysis very easily. And a postdoc of mine, Dr. Vijay Sarthi, who came to me, he doesn't know a single chemical structure. And so since he doesn't know a single chemical structure, the only thing that he can do is to work with the computer and look at sequences, and that's what he's been doing. And another interesting thing about working with computers and looking at sequences, you don't need any money. And therefore you can be retired and still do it. And that's what I've been doing. So here are the variants that you see, the mutations that have happened, for example, in the alpha variant, in the delta variant, and the Omicron variant which you see. There are more mutations in the Omicron variant. I'm coming to the Omicron variant in a moment. You can look at all these mutations, but you can do something slightly different from what you might have seen. You might take the individual amino acids. You know, out of all those mutations, only some of them are critically important. A mutation at the furin cleavage site, the mutation which one calls D614G. Very early on, in the x-axis in all the figures that I'm going to show you, you can't read the numbers. But what are the numbers? Day 1 is January 1 of 2020. So that means if you come to the end of this graph, you've reached the end of 2021, which is towards the end of the pandemic. And what do you see? This is the mutation D614G, happened very early on. By February or March, all American data had only 100% of the sequences had D614G. This one single mutation adapted the virus to human populations. After that, all the other mutations which came only changed the properties of the virus somewhat moderately. The alpha variant swept through America, the delta variant also swept through America. America was one better than India, in the sense that it allowed both variants to sweep through the country. In India, what happened was that the delta variant came early and wiped out the alpha variant very early on. But here you can see the alpha variant going up, and somewhere when we come to June of 2021, June of 2021 was when we were in the peak of the delta wave everywhere. You can see that the co-infections are also possible here. The viral recombination is possible here, but we haven't been able to find signatures of that in the sequence data. The furin cleavage site, where a proteolytic cleavage happens, there are two mutations. There is proline 681 going to histidine and proline 681 going to arginine. This is the delta variant and this is the alpha variant. The blue is alpha, and you can see after some time the delta will take over. I like these because after training in chemistry, if you see a curve like this, it looks like a beautiful titration curve from which you could read a pKa. And you then see that maybe it's almost like a phase transition which happens at some point or the other when the virus takes off. But you can look at infection dynamics by looking at various variants, and won't bother you with that, but except to show you that as you come towards the end, this large spike that you see is the Omicron variant. Now, if I were to go and test all of you, I think at least about 1%, and if there are more than 100 people here, we are likely to find some who are in fact still testing positive for COVID. Then of course you would be the ideal subjects to take some swabs and do sequencing and then find out which mutations you are harboring. But knowing all these mutations allow us to understand antibody responses, and many of these mutations have also probably evolved because of the immune pressure which has been created by vaccinations over this period of time. In the 2020 data, no vaccines were there. In the 2021 data, what has happened is the vaccines have also come in. But America, of course, 50% of the population was vaccinated, 50% of the population remained unvaccinated. All those who were Republicans were unvaccinated, all those who were Democrats probably got themselves vaccinated, roughly. So that's pretty good. But I want to end soon and tell you about the Omicron variant. This tells you the changing face of science. I was supposed to reflect on science. On November 23rd of 2020, there was a tweet which gave the sequence of the Omicron variant which appeared in South Africa. So when I saw this, I copied down all these mutations immediately. But 2 days later, there was another tweet where Q493R, which had been put as Q493K, it had now been corrected. So both the publication of the sequence and the correction both happened on Twitter. So now this is very difficult to keep up with. I'm hoping that Elon Musk will completely close Twitter down so that people can't do this kind of thing. And we will then go back to the scientific literature to find sequences. But these sequences are very interesting. Look at this here in blue. That's an insertion. So you can have mutations, you can have deletions, you can understand them. Insertions are a bit more difficult. Where did the insertion come from? So this is November 23rd. From now on, you have to look at the dates. November 25th. By December 3rd, there's already a paper which says that the insertion might have come, and where could it come? And say this insertion must have come from coronavirus 229E, and I was very happy when I saw this, because that means Dorothy Hamre's virus is still circulating and then recombining, and all kinds of things are happening here. The Chinese, of course, have another take on this. They say it's come from mice, jumped into humans, and so on, but let that be. But science has become almost impossible. I'm sort of glad that I'm retired. And by the 27th of December, already a paper had been submitted to Science which has structures of the Omicron spike trimer with the receptor and an anti-Omicron antibody done by cryo-electron microscopy. So you can see November 23rd, and here is 27th of December, paper published on the 2nd of February. This is not a pace at which we can actually do anything over here, even given all the cryo-electron microscopes which have now appeared in India. So this is a reality that we must face. Lastly, what do you do about viruses? You can't do better than to go back and look at this article in 1988 when the HIV epidemic was raging, and Joshua Lederberg reflected in the Journal of the American Medical Association. He says this: human intelligence, culture and technology have left all other plant and animal species out of the competition. We also may legislate human behavior. This is all that governments try to do. They try to legislate human behavior. But we have too many illusions that we can, by writ, govern the remaining vital kingdoms, the microbes, that remain our competitors of last resort for dominion of the planet. The bacteria and viruses know nothing of national sovereignties. In that natural evolutionary competition, there is no guarantee that we will find ourselves the survivor. And Lederberg was probably one of the most eloquent commentators on the scene in biology, and I think one needs to go back and read him. A last question: where did the coronavirus come from? And this is a question, of course, which bothered people right from the beginning of the pandemic. In fact, one of the very first communications that I got when the pandemic began was from Professor Gupta Sharma here, who sent me a paper which was posted on the archive by a group at Delhi IIT raising a suspicion about the coronavirus. This was, of course, immediately dubbed as a conspiracy theory. There may have been many things which were wrong with that paper, but the question which they asked and the evidence that they produced there was something that deserved much greater attention. The question over the years, over the last 2 years is, did the coronavirus evolve naturally? Did it go from bats to an intermediate animal and then come to humans? Or was it a laboratory creation? This is not an unreasonable question, because the technologies for creating viruses in laboratories exist, and already the laboratories at the University of North Carolina in Chapel Hill, Ralph Barick's laboratory, in collaboration with Chinese scientists, had already humanized the previous coronavirus SARS-CoV-1. So there is no reason that you can't now do those same kinds of things. Effectively, experiments which are called viral gain-of-function experiments, where a virus which infects a mouse can now be made to infect a human cell. When I thought about this, I found the arguments and the doubts raised compelling, but they reminded me of a poem that I'd read in school, written by the poet William Blake. He wondered about two animals, the lamb and the tiger. Both animals, and all animals including us, we have this bilateral symmetry. And when you have bilateral symmetry, sometimes everything looks nice. And so they look beautiful. The lamb looks beautiful. The tiger, of course, looks beautiful, but you wouldn't like to get too close to it. It's also fearsome. The coronavirus also looks wonderfully symmetric, and to some, it would actually, to people like me, it does actually look beautiful. You can even see those trimeric spikes on some of these. It is a beautiful structure. What Blake asked was this about the tiger and the lamb. He says, what immortal hand or eye could frame thy fearful symmetry? Did he smile his work to see? Did he who made the lamb make thee? So, question of course is natural evolution or laboratory creation, and those of you who follow this literature will find that in the most recent issues of the Proceedings of the National Academy of Sciences, these questions are being raised again. Strangely enough, they're being raised by a man called Jeff Sachs, who is an economist by training, but was also the chairman of the Lancet Commission on COVID. So I don't know, there appear to be still doubts. All of the work that I've described to you is done sitting at a desk, never entering a laboratory because I don't have one. And so I was wondering, how do you describe this? One of my favorite authors, John Le Carre, says this in this novel. He says, survival is an infinite capacity for suspicion. And in another place he says, a desk is a dangerous place from which to watch the world. But I have given this last couple of slides in other places. And I gave them at an engineering institution, online of course, during the pandemic. And I didn't know what to say. I didn't know, I couldn't see the audience. Yeah, I can see all of you, that's nice. But the day after I gave the letter, I found this wonderful email from a listener. The listener, it turned out, was a poet. And she is a rather senior poet living in Hyderabad, Syeda Imam. And she said that she liked the chemistry. She said, I guess that is why chemistry has such a magic of meanings. It's actually, chemistry is a magical subject. All of magic is done with chemistry, really, most of the time. You produce things from here, you get things to burn, and so on. And then the next day, she sent me another mail. After a week or so, she said, go and read a blog, which I know, a friend of mine writes it, and she's Dr. Siddharth Mukherjee's mentor. So I went to the blog to read it, and I'll tell you what I found. This is a blog which is based to sort of promote this book called The First Cell. This has been written by an oncologist of Pakistani origin, Pakistani-American origin, Azra Reza, and she's a cancer clinician who's worked for 50 years or so. And she says that she's done this work all her life on treating acute myeloid leukemia patients. And she says she's done this with the same drugs since 1977. And she says that her colleagues treating other and more common cancers are no better off. And she says, I obsess over what I've done wrong and what the field is doing wrong correctively. This is in cancer research on which an enormous amount of money and time and effort has all been spent. But we have to, at some point or the other, even, for example, in dealing with viruses and all, take a step back and ask the question, what is it that we should do in preparation for any future event that might happen, like the unfortunate events of the last 2 years? Evolution is important to understand. And when she titles her book The First Cell, she's really asking a question. She's asking a question, what is it that makes the first cell turn malignant and then grow further? Which is the first cell? This is equivalent to going back to your biology textbooks, to the tree of life, and going to the node there where archaea, bacteria, and eukarya diverge, and ask, what is the last universal common ancestor? How do we approach this? How do we look at the origin of a cell on Earth itself? Here she's trying to worry about the origin of a cancer cell. So I think evolutionary questions are things that must preoccupy people for some time. Lastly, what are the lessons that we've learned from the pandemic? The first one, of course, is that nature periodically provides a reminder of the limits of human arrogance. We believe that we have conquered everything, we can find a therapeutic for anything. This is not true. Nature also demonstrates that the frontiers of science are truly endless. You know, the endless frontier is a famous report given which fueled American science after the Second World War. Frontiers of science still remain endless. The other, of course, more specific. Biology assisted by the chemistry of molecular variation is a formidable force of nature. It is that which has really fueled the coronavirus pandemic of the last 2.5 years. Finally, I must conclude with one acknowledgement, and that acknowledgement is the two institutions which have provided me shelter for almost my entire professional life. The Indian Institute of Science, where I spent 41 years. No one ever asked me there, what are you reading, what are you talking, what are you writing, and so on. And in fact, despite the many things that I said and wrote and so on in the years that I was there, they still made me the director of the institution for several years. And that's a wonderful place. And then, of course, the National Center for Biological Sciences, which has actually provided me a refuge in retirement where I can still go and pretend to be doing a little bit of research. So thank you very much for inviting me, and have a
So now we have some time for some questions. So we can pass around the microphone.
A detail which you mentioned briefly, where in the first letter in Nature, the lady's name was there, but I think later the name was removed. So were you able to find out why that was the case?
Yes, because it sort of turned out in the research that I did, I found that her husband turned out to be also a very interesting person. She married a person who was an associate professor of statistics at the University of Chicago, and he was a statistician, but he was one of the persons who testified in the United States Supreme Court on behalf of the tobacco companies when the connection between tobacco and cancer had been established, and they were trying to bring legislation to put warnings on all the cigarette packets in America. So the tobacco lobby was very strong, and he was actually working for them. So he became a very unpopular figure in the academic circles which don't like the big tobacco companies. So both husband and wife decided to retire. Of course, I must also say that throughout her career, she had only one title, and that was the title of research associate. She was never made an assistant professor or an associate professor, because that was the way things were in the United States at that time, and very, very few exceptions. So they both retired and went away to the mountains in Colorado and lived there. Then, of course, the virologists forgot about her.
Thank you.
We had some conversation during those days about the anonymous scientists. So this is Hambre who we are referring to.
Yes, I have to say that one of my hobbies is to dig.
Right.
She's not the first one that I dug up, but I dug up others. And in fact, it's digging up anonymous scientists is something that can keep you going as long as you can, because there are lots of them.
And we are going to see a glimpse of that tonight.
Yes.
Oh, Mahek.
The Delta virus and Delta variant and Alpha variant. Did the Delta variant evolve independently in India and in United States?
My guess is I haven't yet, I looked at very limited Indian data which was available at NCBI. I think the viruses have in fact evolved independently. And it's not that the virus came from somewhere during the pandemic and then was taken from there. So calling it the Indian variant or the South African variant or the American variant is actually not right. They are the country where they were first identified. That's all it is. Yes. I think there's clear evidence for that now.
In your 41 years of experience, I would just like to know that what's your perspective of virus? Because still in NCRT we see that is it living or non-living. So
I mean, you're asking me whether I think the virus is.
yes, I mean, what's your perspective of
Whether it's a living organism or not.
Yes.
Yes, I think it's definitely a living organism because it reproduces. It only needs the milieu of another cell which it has to infect and reproduce, but it contains all the necessary information for reproduction.
Most biologists believe. I think it's and I think the coronavirus pandemic, we realized, the viruses are the one thing that we can understand now in total atomic detail. Almost at the level where we have structures in the different virus. Maybe not everything in the coronavirus, but only the pieces. But there are others in which the entire, like the rhinovirus structure is known.
So, I have a question. Like when we look at these mutations that are happening in the sequences, as a protein chemist, for example, these mutations, they are not very impressive to me sometimes. I mean, these mutations that are happening, as a protein chemist, I would not consider them as a drastic effect which would really change the structures of the proteins. But somehow they are doing much more beneficial effect in terms of that by the survival or immune response generation or whatever it is. So, what is your take on that? Like these mutations, they don't look very impressive in terms of structure.
See, this is the problem. The mutations are now random. The viruses which replicate faster and faster, like in this case, Omicron, mutations are much more than Delta or Alpha. So even though I put 10 mutations for Alpha and 10 for Delta, you will get Alpha variants and Delta variants which have many more mutations. They acquire them because of the errors in reproducing information. But these are random. Selection then happens. For example, you get a mutation which allows the virus to be more infective, or say in the case of the histidine arginine, arginine certainly gets cleaved quicker than histidine by the protease. So that was there in the Delta variant. So these kinds of things are selected out. To understand viral mutation, you have to just remember Darwin. Variation, selection, adaptation. the virus adapts to the host.
Okay, so I have a question regarding the mechanism of the action of the virus itself.
Can you wave your hand? I can't see you.
So you mentioned that the virus itself binds to the ACE2 receptor, and then a proteolytic cleavage occurs at the fusion site, and finally it's followed by the fusion of the membrane. Yeah, so what is the difference between SARS-CoV-1 and SARS-CoV-2 in the mechanism of action? Because certainly there seem to be various differences in the effects they produce in the human body.
I don't think there's any major difference in the ways in which both viruses infect cells of the respiratory tract. Both viruses use the angiotensin converting enzyme receptor. So they both have the same mechanism. That's why work could proceed so fast. As a sidelight, I might tell you that the Dorothy Habray's 229E coronavirus, about 30% of our common colds are coronaviral colds. They use not the angiotensin converting enzyme, but they use the leucine aminopeptidase as the receptor. See, human cells or animal cells have a lot of proteins on the surface, many of them proteases, which are used for various other purposes. And the virus just uses them opportunistically to get into the cell.
Okay, and in one of your slides you showed a graph of the alpha and the delta variants, if I'm not wrong. So there was a point of contact between the two graphs where one decreased exponentially and the other increased.
Yeah, I was most interested in that because at that point you would imagine in America, about half the people who were being tested were infected with the alpha variant and the other half were being tested infected with the delta variant. It's also possible that there were many people who had both variants at the same time. If you had both variants at the same time, then there is the possibility of a viral recombination producing a new variant. Recombination events could produce much more mutations than you imagine. But there doesn't seem to be evidence for it.
So how do we justify that one strain wiped out the other due to the pressure of selection?
No, because these mutations in the alpha and delta variant were actually exclusive, except D614G, which is there in all variants. The other mutations.
Recently, just now mentioned that mutation D614G, that is the one that resulted in adaptation of the virus to human population, correct? So at what point did this mutation occur? Like previously, was it the case that the virus wasn't adapted to human population?
When I say adaptation, D614G helped the virus to spread rapidly in human populations. The original Wuhan sequence, which had D at 614 position, also was an infected virus. It did spread. But if you look at the data, you will find the very early sequence data which was, see what happened in the early days of the pandemic was also this, that everybody was not sequencing everything. In fact, there's much more sequencing going on in 2022 than there was in 2020. I had to gear up. But in the early sequences coming from January to March of 2020 in America, you find that the Wuhan sequence predominates for the first few weeks. But very quickly, it's completely swamped by 614G. This was known in the literature. Everybody pointed it out very early on.
So what is the reason behind a particular strain or a particular mutation being completely or maybe majorly spiked off from infecting? Like, at what stage or what are the reasons behind a particular strain of virus being spiked off? What results in that really quick effect?
I did understand your question, but I'll tell you one piece of information. The coronavirus spike protein is a fairly complex protein, very big protein. It has very clearly segregated domains. and there is an N-terminus domain of about 300 amino acids, right from the N-terminus, which does not have any role in infection and all. But the major mutations which have driven the pandemic appear to be deletions in the N-terminus domain. Deletions of bulky residues. But what is interesting is the deletions that are found in South Africa, the deletions which are found in India, and the deletions which are found in Europe and America are different. And it would almost appear like that the initial deletions which happened in the coronavirus were somehow driven by the immune pressure of the antibody response that was mounted by the populations which were infected. Now, the antibody response is also under genetic control. So, different ethnic groups probably mount different antibody responses. I think we are going to learn much more about the evolution of the coronavirus spike protein in the years to come when all this data is looked at. Now, there's a very old statement which says when there is a disaster, you should wait for some time before you analyze the cause of the disaster. And this was not said with respect to viral pandemics, it was said with respect to the economic depression of the 1930s in the United States. So even economic depression is better to wait. After everything come back to normal, you go back and look at your data and now ask questions.
Thank you, sir.
Another question. Of course, while talking about chemistry and biology border and interaction, it reminded me of one slide that you displayed more than 20 years ago at IISER in a lecture. Dodson's quote on chemistry that's an engine driving biology.
Yes, I guess people say that chemistry is the engine drives biology, but as far as research is concerned, today I would say it's biology which is the fuel which powers chemistry.
Right.
So every chemist will be working in biology except those who have changed their faith to material science.
All right. So, IISER Mohali, it's my pleasure to give you a little memento.